Good evening, everyone. My name is Takiwa T. Smith, founder and executive director of Science, Engineering, Mathematics Link Incorporated, and I want to welcome you to this evening's Teen Science Café. Our Teen Science Cafe is a program of our Math and Science Career Academy, whose goal is to expose youth to STEM and STEM careers by connecting them with various members of the STEM community to learn about STEM careers, as well as engage in and view interactive demo and hands on activities when we're able to be in person. The purpose of our Teen Science Cafe is to provide opportunity for teens to really take an in depth look at a particular STEM career by learning about the speaker's STEM journey, what they do every day, and having an interactive demo video that shows a day in the life of their work. The reason we chose to focus on teens is because this is a critical time in your K through 12 educational experience where you're making decisions about what you want to do when you graduate from high school. If you're in middle school, you're deciding what STEM clubs to participate in, and if you want to go to a special
Mhm.
magnet school to study a STEM field that you're interested in. If you're in high school, you're also selecting what STEM clubs and summer research programs and what you want to major in college. So these are critical times for you to make decisions. And it is great as you're making that decision to have someone that is doing something that you're either interested in, that you kind of are interested in, or that you didn't even know about. But by listening to this speaker and learning about that career field, you were learning, like, hey, I can do that. And this person is showing me how. And so we hope that as you watch this cafe, that you learn a lot about this particular career path that can help you shape what you want to do when you grow up and help you start your journey to become a STEM professional. So without further ado, I'm going to introduce our program coordinator, Carlin Pounders, that is going to introduce today's speaker.
Thank you, Tequila. Hi, everyone. Thank you so much. As was mentioned, I am Carlin, the program coordinator, and I have the awesome pleasure of introducing tonight's speaker. So Jennifer Phillips earned a Bachelor's of Science in Biology from Indiana University, Bloomington, a PhD in Molecular Biology from University of Oregon, and completed a postdoctoral fellowship developing zebrafish models of Usher Syndrome, a hereditary form of deafblindness. As a member of the research faculty at the University of Oregon, she uses zebrafish to learn more about diagnosis and treatment of rare human genetic disorders. So I'm really excited that we get to talk about this topic for October. And without further ado, I will hand it over to Dr. Phillips.
Thank you so much, and I'm really pleased to be here. I am one of my favorite parts of my job is talking to young people who are trying to decide what they want to do, if they want a STEM career, and if so, which track of a career would that be? So I'm really happy to be able to share my journey, and I'm going to tell you a little bit about my work, how I got here, what I do, and hopefully there'll be an opportunity for you to ask questions about anything you want to know more about, or at least just think about things that appeal to you or things that sound like things that can help you decide where you want to end up. So I am working at the University of Oregon as a research scientist, as Carlin introduced, and I'm in the Institute of Neuroscience is where I do my work. Neuroscience sounds a little technical and to some people scary, and it kind of makes you think of brains, um, which is not always the case. Lots of people do indeed work on brains in neuroscience. I'm not one of those people. I seldom think about brains. There is plenty of other neuron activity to talk about and think about in the realm of this field. And this encompasses a lot of different types of biology and also psychology. When you're talking about neuroscience, there are some very kind of base level biophysics and biochemistry things that go on when trying to understand certain aspects of neuroscience, genetics, evolution, molecular biology. the way that things develop, cell biology, and then also behavior, and psychological and brain output choices that are part of that as well. Now, I don't focus on all of those things. My realm is pretty much biochemistry, genetics, molecular biology, and cell biology, so a subset of all that. Um, and while I'm going to be talking about our work with the animal model system of zebrafish, um, not everybody is feeling okay about working with animal models. So I do want to stress that in this field and also many others, there are many opportunities to do really impactful work that has nothing to do with using an animal model. There are lots of computational analysis styles of neuroscience, building the machines that detect things without actually doing the detection yourself is a valid choice. There's a huge amount of machine learning going on right now, which is taking concepts from neuroscience and also the field of mathematics and physics and psychology to develop things like artificial intelligence and that kind of stuff. So many different options for where to go, even in this one institute alone. Um, and what I am working at is trying to give you a sense of the range of what this field is, just in general, not just neuroscience, but in general, this kind of hands-on lab research. Um, Most of the time, people at a research university that's not attached to a medical school or something like that is working at the level of what we call basic research. So this is just knowing things for their own sake, the power of finding out facts about the natural world because it's satisfying to know the answers to those things. Um, on the kind of the other end of that spectrum, there is applied research where you're taking a collection of known facts and creating something that has utility in technology and medical in industry, anything like that. And then there's kind of an in-between space that we call translational research, where you're taking concepts from basic research and figuring out, do they apply to something in some of those fields, and if so, how? But also taking a need from some of those fields and figuring out, is there basic research that needs to happen in order to connect these dots or close these gaps in the applied research world. So I do basic research, and I also do a little bit of translational research in the work that I do, making animal models of genetic diseases, just to know where I'm starting from. So I was asked to talk about my road to where I am now, and I will tell you up front that it's a pretty windy road. It was not a straight path at all. I wouldn't necessarily change anything about it, but it was a circuitous way to get there, and I want to tell you a little bit about that and some of the reasons why and what I learned along the way. So growing up, I was always very interested in the natural world. I love to be able to build things, to read about science and nature in any way I possibly could. I didn't have There was no such thing as a Teen Science Café in those days. We didn't have a whole lot of opportunities to actually even take that many science classes in school. And I didn't have anybody in my family who was a scientist. Most of the people in my family didn't really understand why I was interested in the things that I was interested in and not their careers, which were decidedly non-scientific careers. So I was kind of finding my own way, like, this is what I'm vaguely interested in, but I don't even know what to call it yet. So I went to school at Radford University in Radford, Virginia, and I kind of wanted to maybe study psychology. I kind of had an idea that, some kind of something in a lab was cool. I love to mix things and do that kind of stuff. And there was a major called medical technology that sounded like that would enable me to do something in a lab. And so I started on that, and that was all going okay. And then unfortunately, some events happened in my personal life that that really derailed that whole thing. There were a lot of things that happened kind of in close succession that were just too much to manage, and so I had to leave school at that point. So a couple of years went by. I found myself over the course of that time in Nebraska, at the University of Nebraska. And in between leaving Radford and winding up in Nebraska, I had worked at some veterinary clinics as a front office receptionist, and veterinarian, seemed like a really pretty cool job. So I was like, okay, now I'm gonna take pre-vet classes, and I'm gonna go to vet school. So probably this is true in a lot of veterinary programs or pre-veterinary programs, but particularly in the Midwest, in a place like Lincoln, Nebraska, there's a very heavy emphasis on animal science. So a lot of the classes that I took actually took place in barns. It was kind of stinky. There was a lot of pooping. And it was really not what I had hoped for in terms of getting me on to where I wanted to be. At that point, I was really questioning my choices. I I had a lot of debt, and I didn't have any money to pay for it, and I was questioning, is this the right thing for me to be paying tuition for? And the answer was no. So I left that program behind. I spent a little bit more time out of school. I worked. I paid my bills. I figured out, this is a better direction to go in. I wound up in Bloomington, Indiana, at Indiana University. And then I was back to, I'm just going to get my biology degree. Everybody I know who's gotten a biology degree has gone to medical school. So that's what I'm going to do. So I was, at that point, I had enough accumulated basic college classes that all I pretty much had to do was just take biology classes. And also French. So I had a lot of time in my schedule to take lots and lots of different biology classes, upper level biology classes, which were all just fantastic. And all of my classmates, probably, I would say probably like 90% of my classmates were pre-med students. And I started to notice a couple of things. I noticed that I was enraptured at learning new information about how things worked in biology, in cells, in molecules, in bodies, and all of that stuff. I was so keen to know the details of that. And my pre-med classmates, by and large, were not. I would come out of a lecture going, "That was so cool! Did you hear that?" And they would be like, "Yeah, I'm just going to get an A on the test and forget all about it." They were approaching this as it was a means to an end. They needed this class. They needed to get the grade in this class so they could go on and do what they really wanted to do, which was to be doctors. So they were not having the same experience on the journey as I was, and that made me think, that this is a difference. This is a big difference for me. So another thing that happened was, because I was such a keen student who loved to know how things worked and what the origin stories of everything was, one of my professors noticed my extreme geeky eagerness and said, I think that you should investigate working in a research lab. There's a lab here that does a lot of the stuff that you ask questions constantly about, and you should go and meet them. So, and I had no idea that that was a thing that a person could do. I didn't really think that research labs, I had only the vaguest idea of what that even was, let alone that it would be accessible to someone like me. But I went up to the third floor of the building that I had all these classes in, where lo and behold, there were many labs. And I walked in and I felt like I was home. This was a place where people were doing exactly the thing that I had always wanted to do, and I that was decided. So I was able to understand what I really wanted to do, which was at this level of basic research again, the power of knowing these facts about the natural world, because it delighted and inspired me to know these things and to find out these things and to understand that everything in the textbooks that I had been reading up to this point were discovered by people that were working in these lab situations, and I could be such a person. That was delightful to me. So That led me directly to, I want to go to graduate school, I want to get a PhD, and I want to do research. That's my life. That's what I want. And from then on, it was pretty smooth sailing. I was able to get into a PhD program, finish my PhD in a reasonable amount of time. Then I got this postdoctoral fellowship, which you've heard about, and now I have my current appointment doing what I really love to do. So what you can take away from that in terms of life lessons, if you will, are the things that I learned at these junctures on my very windy road, is to be able to find and identify your strengths, be able to actually name what your passion is. And sometimes it takes experience to be able to know, to even get that vocabulary. I had this feeling of what I wanted to do, but I didn't know what it was called because I had never seen it before. And when I saw it, I knew it. I could identify it right away, like an old friend, because it had always been in me. um, Another thing that I would recommend to tap into is just to know what a good challenge feels like and how to differentiate that from horrible drudgery that you're not getting anything out of. A good challenge feels like it's hard work. Sometimes it's really stressful, but you know that doing the work is going to lead you somewhere good. And then the last thing I have on here is to find your people. So if you're going into a graduate level research program, then you're going to be suddenly surrounded by people who have really similar interests to your own, maybe for the first time. Suddenly, you're not the outlier anymore. You're with people that are just as excited about that as you always have been. And you'll be able to talk about things at a depth that you haven't maybe been able to have conversations at before. At a certain point in your training, you're going to know a lot more about a thing than most other people in the world do. And that makes conversations sometimes challenging. You're kind of the killer of the party conversation when people say, "What do you do?" But if you find your people, you can have those conversations with them, and it's all good. And then you can work together, you can collaborate, you can figure out new ideas and new way of doing things, and that's all fantastic and inspiring, and definitely part of the journey that will help you get to the point where you can thrive. So now I'm going to segue to talking about the zebrafish that I use, and what a question that a lot of people ask me when I do these sorts of things is, why zebrafish? It seems like a pretty random little unrelated thing that most people have never even heard of before. So what I'm going to show you in the next few slides are what a fish can actually teach us about how our bodies work, how their bodies work, how genes are affecting our lives. And I'm hoping that you're going to have some questions about that at the end. And I hope that I will be able to tell you how incredibly important and useful zebrafish are to us in learning all of these things. So just to break it down a little bit more, when we're talking about genetic diseases, are talking about things that originate in the DNA. DNA in all of the cells in the nucleated cells of your body have the creation and maintenance instructions that you're going to use for your whole life. But what that actually means is that they are controlling the things that happen in all of your organs and systems. And as you're developing, as you're growing into adulthood, and as you're functioning as an adult, and as you're aging, and as you're potentially reproducing, and all of those things, there's a whole lot of genetic regulation that goes on in every step of the way there. So while I will be careful to say that your genes do not define you, there are many other things that impact the person that you are, and how and your health and your lifestyle and everything. The genes definitely inform all of those things. So when we're talking about looking at human diseases, we hit a problem almost immediately, in that humans are extremely variable in what their specific genetic recipes are. There's a lot of differences from one human to another. No matter where you come from, or anything like that, we have a lot of variation in our genes. So sometimes that makes it pretty difficult to find meaningful messages in the DNA code. That's one reason of several why model organisms that are non-human are appealing to us. And when a researcher wants to study something that has bearing on human health, but it's not possible or practical to use humans, then they're going to turn to a model organism, and they have a choice to make about which type of model organism they want to use. So I kind of have a little mini spectrum of model organisms here, where on one end you have animals that are similar to humans. So there's an advantage there in that if you learn something from an animal that's not human but is genetically similar, very similar to a human, then it makes it more applicable to whatever question you're asking about human health. The downsides on that side of it is that those animals also tend to have a lot of genetic variability that makes them just as messy and sometimes difficult to study as humans are. Their generation time is pretty slow, meaning how long does it take them to grow up, be of reproductive age, and actually produce offspring? And then when they do that, their number of offspring that they have at a time is a pretty small number. So that makes it hard to have really powerful statistics if you only have a very small number of things that you're able to work on. It's also quite ethically controversial to work on some of these animals, particularly the warm and fuzzy ones, the large animals. A lot of people are not comfortable with that. That is completely fair and respectable opinion to have, and that would certainly all influence someone's choice over whether they wanted to choose an organism on that end of the spectrum or not. So at the other end, you have things like the tiny worm that lives in the soil called Caenorhabditis elegans, that's its Latin name, or the fruit fly, which is obviously not to scale on this slide. Drosophila Melanogaster is the normal lab option. It's such a tiny little fruit fly that comes into your kitchen in the fall and crawls into all your apples and so forth. These guys are very easy to work with genetically. They have a generation time of days where you can go from being an egg to making your own eggs in, so 72 hours or something crazy fast like that. Nobody really has a problem with how many of these animals are used for research. They're not regulated in any way because, nobody has, very few people have concerns about fly ethics and that sort of thing. But the downside is, of course, they're fairly dissimilar to humans. There are a lot of parts that we don't have in common. There are a lot of genes that are not conserved between their genome and ours. So some of the things that you would want to ask in a human can't be asked in these organisms because they just don't have the equipment. So I'm biased, of course, but right in the middle of the spectrum, I've seated the zebrafish because the zebrafish has a lot of the features of both and some of the minimized concerns. The zebrafish is really quite similar to humans, both genetically and in terms of their body patterning. Clearly, there are some obvious differences, but we have about 75% of our genes in common. And if you ask that question with how many genes in common do we have that are related to human disease, that number goes up to about 85%. So there's a good chance you'll be able to find a gene that you can work with that would have some bearing on human health. And I'll tell you a little bit more about the power of the zebrafish in these next slides. So one of the things that we love about them is their transparency. They have this beautiful optical clarity under the microscope. And when you set up a breeding event between a female fish here, see her white belly there, and the male fish, they will fertilize fertilize their eggs in the water, and there'll be hundreds of eggs that are just developing out in the open. They don't develop inside the mother's body or anything like that. So you can see them and watch them develop almost immediately. The eggs are actually transparent. This is a one cell stage fertilized egg. The actual cell is this clear little bubble on top, and then underneath is a big sac of yolk. That's what's going to feed the embryo as it grows throughout the first few days of life. And the time up here, this HPF stands for hours post fertilization. So this is 15 minutes after fertilization. You have a nice big cell on top of the yolk sac that's developing. And then by an hour after fertilization, you have 4 cells. And then 15 minutes later, you have 8 cells, and so on and so on. So by the time you're at 19 hours post fertilization, so not even 1 day of development, um, you have something that pretty much almost looks like a fish already. It's still completely transparent, but here's an eye, here's the tail. It's starting to get a little tail. In a couple more hours, like 22 hours post fertilization, it's going to have enough nerves and muscles developed that it can actually start swishing its tail around. Here's the yolk sac. It's a little bit smaller because it's consuming it, but it's still powering that embryo with nutrients until it is big enough to get its own food. Um, by the time the embryo is 2 days old, it's got these little pigment spots. Even though adult zebrafish have stripes, and that's where they get their name from, obviously, the little guys have spots, these little dark spots on them. Here's the yolk sac again. And this embryo is inside an egg sac, which is called a chorion, which is also conveniently completely transparent, so we can see through at that. You can make a little hole in this egg sac and take the fish out. It doesn't hurt it at all. If it's in buffered water, it's completely safe and happy to be out of the chorion. But that's how it looks if it's not all curled up in there. So very fishy already. It's got a heart that's beating, and I'll show you a video of that in a little bit. And then by 5 days, this is 120 hours post fertilization, or 5 days old, you have this animal, which is still very tiny. These are about a millimeter long or so. But it's got sensory organs. Here's an eye, here's an ear. It's got a beating heart. It's got a mouth that can feed. It's got a gut that can absorb and expel and disseminate nutrients. It's got muscles and nerves and a big, beautiful brain. And it can do behaviors and all these different systemic things in the water that enable you to study it in a very short period of time. So inside of a week, you can have thousands of these guys swimming around to be able to observe whatever the experiment calls for. Another really cool thing we can do because of that optical clarity, that transparency, is to label different proteins. And I just have a couple of things here just to show you. These are all live. These are pictures of live zebrafish larvae, and I have the ages down here. Here's a muscle protein that you can see growing and developing in the muscles of the trunk and the tail, and also a little bit here in the heart. This is a protein that's labeling the blood vessels of the fish, which you can see going all through the gut, through the trunk and the tail, the brain, the eye. There's the heart. And then, shout out to the Institute of Neuroscience. Here are 3 different kinds of neurons that you can see are occupying different regions of the brain. Here's one little blue set, red set, and a green set. And they're also occupying different regions of the eye. So different cell types in the eye, which also contains a lot of neurons. So this can give you an appreciation for the complexity of how these things are put together, but also how wonderful it is to be able to see these things in a live animal and just watch it grow, watch it develop, watch those patterns establish themselves and change. That's a really powerful thing with the zebrafish. And then we can also use different chemicals to stain body parts like the skeleton. A lot of the diseases that we work on have symptoms that involve changes to the way that the bones of the face are formed, or the bones of the spinal column are formed. We can look in the fish at different stages and see if we can see similar symptoms coming up in those structures as well, either the face or the spinal cord. And these are all fish that are developing quite normally, but it's just to show you, to illustrate the types of things that we can see with these guys. So I am going to show you a little video now. It's a little bit cringe for me to watch because I'm in it. And but it's me and my boss, Monty, talking about some of the projects that we do with the zebrafish. And it'll give you an opportunity to hear a little bit about that, but also to actually see the zebrafish facility, where the place where we keep the fish, how we put them together and collect eggs and do certain things like that. So I am
You're ready
going
for me to
to,
start it?
yeah.
Or do you think you want to try it or no? Okay.
It's not going to work in the PowerPoint. So
Okay.
if you
I
can
will.
switch over to the other thing, there we go. Awesome.
The main focus of our research is to study Usher Syndrome, the leading cause of combined deafness and blindness in humans. What we've done thus far is to take the exact sequence that's present in the human gene and that is different in patients that have the disease, and we've introduced that sequence into the zebrafish gene.
The zebrafish develop their visual symptoms very early in life, so we can see things start to go wrong within the first week. We can then use those fish to understand and learn how to treat the disorder. One of the vision tests that we can do on young zebrafish is this apparatus called the Optokinetic response assay. And when the drum rotates, we can record how well the fish can see the moving stripes. It's basically just an assessment of how well can you track a moving target. So sometimes fish will perform very well in bright light, but not so well in dim light, which is consistent with what people affected by Usher syndrome experience.
The support from the Usher Collaborative is absolutely critical for the work that we're doing.
Every Usher 1 fish we have in existence right now is due to the support that we've gotten from the Usher 1F Collaborative. So we've been able to make a number of different zebrafish strains that carry particular genetic mutations that can help us understand how this gene works. Now that we have developed and characterized the Usher 1F models, what we hope to do is be able to expand the potential treatment panel that we're using. That information is going to help us be able to evaluate our particular treatments working. The way in which the field is moving forward with the discovery of how to really fine-tune these genetic changes is something that all of humankind has a stake in.
Okay. Let's see. I'll be able to share that. Okay. Thank you, Tokyo. That went pretty smooth. Whoops. No. Okay. So that was kind of the packaged deal of how we were able to do that. I thought I'd take you through a few more details on how we are actually able to create a zebrafish model of a human genetic condition. This would apply to the things that we just talked about with Usher syndrome, and I'm going to give you another example here in a few minutes of what we actually do. And this is pretty nitty gritty. I hope that people will ask questions about it if they have that. And because there's just a lot encompassed in here. So most of the human conditions that we work in are conditions that can travel through the genes in a family tree, and may not ever be manifested until you have two parents who are what we call carriers. They have the little orange gene is something that, if it gets together in the right combination, could cause a particular set of symptoms for one of their offspring. And as you can see in the depicted offspring down here, there are various ways that those genes could be inherited by these 4 children, and only 1 out of the 4 happened to inherit both of the orange genes, and therefore is showing the symptoms of whatever this thing is. So there are a lot of human diseases that we already know and understand very well about the cause of that. If you have a patient who's appearing in the clinic and has a certain set of symptoms, it might be pretty obvious, or you have a strong suspicion of what that is, and then you'd want to confirm it by taking a look at their DNA sequence to see if indeed the gene you suspect is affected in that. And then that's called a genetic diagnosis. That person has a condition that has a genetic identity, and you proceed accordingly with whatever the standard treatment is in that situation. However, if the disorder is more unusual, if it's rare, if it even hasn't really been seen in that presentation before, it's not that easy to make the call. You would do some DNA analysis of the patient and hopefully of their family members too, to try to get a sense of what was going on there. But at that point, it becomes incredibly useful to use a model like a zebrafish model to help solve the mystery and to make that genetic diagnosis. So what we would do in a situation like that is to choose the zebrafish gene that corresponds to the gene in question, the candidate gene in the human family, and then inject some chemicals that are going to manipulate that gene to convert to a form that's in that affected individual. And this is one of the things that you just saw me doing in the video, is to being injecting those little tiny bubbles, which are actually the zebrafish embryos. So we can inject that. It's not going to harm the fish or alter the development in any way. It'll just grow up having some number of its cells of its body changed in the genetic way that we are trying to make. So then we would breed those injected founders once they get old enough to a fish that doesn't have any genetic differences at all, and then get that big pile of offspring to work with. When we have a bunch of little baby fish embryos, then we can take a subset of them and sample their DNA and mix them in a tube with some building blocks of DNA and some enzymes to make the building blocks go together, and look at that gene of interest to see, did we make the right change? Do we have the desired mutation? Do we have the desired genetic variant that we were wanting to kind of mimic from the human patient? This is a process that you might have heard of called PCR. That stands for polymerase chain reaction, which basically just means you cycle through a bunch of building the specific little part of the DNA with all of these ingredients until you end up with enough DNA that you can run it out on a gel. So it's like a kind of a slab of gelatin that's got holes in it. You can put the PCR samples in the holes, and because DNA has an electrical charge on it, you can use an electrical current through that gel to separate the pieces of DNA by size. This is a picture of a gel that's showing you different sized fragments, and you can tell the genetic identity based on what you put in. So this is how we would identify which of the founder fish that we injected and then grew up to become adults are carrying what we're interested in doing. Those are the ones that we are going to keep and propagate. We're going to breed those guys together until they get big. And then when we can cross two of them together, just like the human family, where the two parents were able to make offspring, we're going to do that and come out with the same result, where there's some number of their progeny, or 1/4 specifically, are going to inherit both copies of the change that we made. And then we're going to be able to compare those fish to the human family and see what kind of symptoms are like. What is the zebrafish going to be able to tell us about the genetic underpinnings of that condition? So I'm going to tell you guys really quickly about one sample. This is a project that's still in progress, and I actually haven't ever talked about it to anybody except for my very small research group before. So this is part of the group that we work in the research group. It's called the Undiagnosis Diseases Network. We have a group of doctors and geneticists and other researchers with different skill sets that kind of come together and work on these really rare cases. And we all do different parts of the puzzle and then put it together to see what we can get. In this particular case, we have a family who have 4 children, and 3 of them inherited one bad copy of this gene from each parent and have a similar set of symptoms. So I'm going to call it the T gene. All of the 3 offspring that inherited it happened to be brothers. That doesn't necessarily mean anything. It's just the random luck of genetics. And they all have the same set of symptoms, which include anxiety, an abnormal heartbeat, and a risk of early death. In fact, one of the brothers of this group died at age 17 because of this heart abnormality that they have. So the question that's put to us is, we think we have this candidate gene, the T gene. It looks like that's probably the cause, but what do we know about it? How can we find out and demonstrate what it's actually doing in this family, or indeed in any other person who happens to have these mutations, who may be born in the future or may just be undiagnosed somewhere else in the world. And that's what we're going to do with the fish. So we are we went through the process of creating a line of fish that has similar genetic changes to what those three brothers have, and we looked at their heartbeats first of all. So we're looking at the rate of the heartbeat, and you can see it in the chest. There's two zebrafish hearts have two chambers here, so you can just kind of see that very rhythmic pulsing, which of course we can count how many beats per minute, just like you can in a human patient. And then with the 3-day-old mutants, I want to hide that. Oops. What happened? Sorry. Okay. There we go. So by having both of these videos play at the same time, so you can see the difference in the rate of the heartbeat possibly, but also the fact that there's some abnormality around the heart. There's some swelling of the sac that's around the heart. That's probably going to cause some problems and is due to some defect in the way that the heart is patterned. And I know it's probably difficult to keep your eyes on both of those at the same time. And I'm not going to show you a lot of really hardcore statistical data, but we filmed a number of these side by side and then counted the beats per minute. And these are much faster. The fish that are emulating the genetic change in the humans have a much faster heart rate, which is similar to the symptoms that they have. So we have a lot more work to do with these, but it looks very promising in that we do have a model of the disease that we can now use to ask more specific questions about what's going wrong in development. How is this affecting the heart? Is there anything that we can do about it? Is there a treatment that would make sense based on the molecular basis of what's going on in the first place? Lots of things like that. So it's a team effort. The zebrafish are a small but very important part of it. And this is the kind of work that is pretty gratifying to do after all this time. And again, it brings us back to knowing facts about the natural world, of just understanding what this hitherto unknown gene is doing to make some very important decisions about the development of our hearts in a normal situation that is apparently going wrong when there's a problem with that gene. And then how we can translate that basic research into maybe doing something for the people that have that on the clinical level. So with that, I will thank you very much for your time. And I'm hoping that I'll be able to hear some questions about anything you want to ask that I talked about, or if there's something that you wish I had talked about that you want to ask, I'm very open to that too. So thank you so much for your time. Oh, can I hear?
Nope, I was on mute. So that was really cool to see, particularly the work that is done in the lab and the equipment that it looked like you guys use.
Mm-hmm.
It looked pretty high tech to me. So a question that I did have was, as far as the T gene.
Mm-hmm.
From my understanding, you guys, as you said, are altering this, like the letter T to it is just a letter applied to it, but you really don't know what gene it is?
Oh, I do. I just didn't want to say it.
Oh, okay. No, that's
Yeah, no, we know it's a gene that's named and it has a known sequence and known identity. It's just gene names are not very. I mean, it's not like it's not a secret. It's just a really unwieldy thing to say. It's called tax1 BP3. So
yeah.
as it not saving myself, saying tax1 BP3 over and over again because it's not, that's not the important part. But yeah,
So
we know the identity of the gene. We just haven't. It hasn't ever been directly connected to human disease before, and that's what we're trying to establish.
So when students are, looking to study these different parts of DNA, these genes, what would you say that they can expect to, how much work can they expect to put into learning these things? Because it sounds pretty intense. and we know that STEM fields a lot of times can be challenging,
Mm-hmm.
and you said so yourself. But just to give them an idea of how much work goes into basically gaining all this knowledge.
Yeah, I mean, it's years of study and, identifying the part of the field that you're most interested in, that you're going to go the furthest in and know the more about. The benefit of having these collaborative research groups is that we all don't have to know all the things at the same time. We can put together a group of people that has different areas of maybe slightly overlapping expertise. So that just kind of maximizes the skill sets that we have. But I mean, in terms of how many years it takes to get the credentials, it typically 4 to 6 years of a PhD program, and then another, I would say, 2 to 4 years of a postdoctoral fellowship. Not all STEM fields require a postdoctoral fellowship. Many do not. So it just depends. It's like a residency. It just gives you a special skill set that you want to develop to further your own, expertise and career. And I did also want to say that in a lot of, especially the natural sciences graduate programs, there's no tuition cost. You don't have to take student loans out to do those things or anything. They actually pay you a stipend to go, and they cover your tuition and pay for you to go to conferences and things like that in the majority of situations. So that's definitely was a consideration for me in terms of what I was able to manage in terms of a post undergraduate experience. So yeah, that was one thing I wanted to mention.
Awesome. And so another question is, you mentioned earlier your pathway to where you are now. What was your motivation as you maneuvered through the science field? And I feel like that's a good question to follow up with everything you just talked about.
Well, I mean, overall, my motivation has always just been to never stop learning. I just love to know new things. And being able to be in a situation where when I learn something new, there's a good chance that, at least for a little while, I'm the only one in the world who knows that thing until I run out of the microscope room or, write the report to tell somebody else. Then that knowledge is mine to kind of, rejoice in privately. And that's cool. I that never gets old for me, being able to just look into the microscope or see what story the fish is telling me, and then be able to share it as part of our community, our global body of knowledge. So that's always been a big motivator. I think it's just the the power of knowing things. And then, being able to help people, being able to have families who are feeling grateful that somebody cares enough to work on the problem. Somebody wants to understand what's going on in their child, where maybe, they haven't always met with that kind of openness and acceptance in their medical journeys. So that part's really important to me too.
Oh, yes. Another question is, so I am of the opinion, now I love just as you described, the camaraderie and collaborations that I've seen from the STEM community, but I'm of the opinion that a lot of times people that are outside of STEM don't really understand. They don't understand it, so they're less likely to take interest of it, and thus almost kind of judge people who are in the STEM field. And I say that to say that, particularly for women, girls, people of color, kids of color, sometimes they can run into instances where it's a little bit of a challenge for others to either take an interest in what they're talking about, or even believe that they are, experts of the STEM subject that you're talking about. So my question is, what advice would you give to students who are either, girls, young women, or students who are kids of color, who might run into some challenging situations when they're very passionate about STEM, and they might encounter someone who makes it difficult for them to do whatever they're trying to do in the STEM field?
Right. Yeah, that's a great question. I did not have a whole lot of support when I was doing it, and I think that probably one of the biggest keys to my success was not only that I had this drive, I had this passion about the thing, even though I couldn't name it. I really felt strongly that that was what I just kept finding my way back to that road because I really wanted it. But probably it would be fair to describe that as stubbornness, so I guess then my advice would be, if you want that thing, then be stubborn about going to get it, and you know finding good mentors and finding people who do get you and who you can talk to about that thing and will give you some honest guidance and career advice is key. But sometimes those people are pretty thin on the ground, especially if you don't see yourself represented in the field that you want to get into. And I mean, I'm super happy where I am, but I know that the STEM field has a lot of problems with not being as accessible to everybody as it is to a pretty narrow demographic. So sometimes you have to be your own mentor. You have to do enough just kind of affirmative training for what you want, and coach yourself into it. So yeah, be stubborn and do the thing that's calling to you.
I love that. And so I would say my last question for you, Dr. Phillips, is if students, after watching this, are like, I want to do that work, I think it's awesome, what do you believe that they can do right now in order to just get them on that path?
Right. So if there's a student who's in middle school or high school, um, finding things that pique your interest in the classes that you have to take as you go through is really important. If you're particularly interested in physics, if you're particularly interested in chemistry or in math or whatever, getting a little bit deeper into those subjects as you go gives you an opportunity to learn more about not only the findings, the things that we do in the lab or the things that people do in their STEM research are what ends up in the textbooks to begin with. So what you're reading about as a high school student are things that somebody experimented on and did. So understanding not necessarily the people that did it, but the process of finding out that information would be something to hone in on. And then once you're in a university situation, finding all the opportunities you can to get some research experience, whether that be getting a paid research position in the lab or taking a class that has some research elements to it. If it's not possible to work in the labs, there's different availability of that. But the more hands-on experience you can get with the thing that you think you want to do before you're absolutely on to the next stage will really help you refine your preferences and make a lot of things clear.
I love that. And I love the mention of paid experience. I would be remiss to repeat again that Dr. Phillips mentioned that there are great opportunities, particularly when you're in university, to attend conferences, you said, and do grant research, I believe you said. So those are definitely good takeaways.
Yeah. And if you know that they exist, then you can start asking about them and start finding out what are the opportunities where I'm going, what kind of scholarships are there that would support research, what kind of paid research opportunities are there. You have to know enough to ask, and now you know. So go, get in there and do it when you have the opportunity.
Well, Dr. Phillips, we want to thank you so much for making your time. This is one of the blessings of the pandemic, because we would have had to get you to a flight to Atlanta or DC. But now, we can have you share your cool research on fish. We were saying before the started that I'm Southern, like I'm only see fish fried. So to actually see that fish, or important animal model in disease research, I would have never imagined that. So thank you for sharing that. Thank you for sharing your career journey. Thank you for especially being so transparent, right? Because we are so taught and ingrained that there's only a linear path, right, to get where we want to go to. And we have to have it all figured out by the time we graduate high school. And we don't, right? We can always change direction and learn new paths, right? So thank you for reminding us of that and reminding teens of that. If you need to change directions, it's okay, right?
It's okay to not have it all figured out when you're 18. I promise you, it will come in its time.
So we thank you so much for giving time, sharing your journey. I learned a lot, and I'm sure the teens that are watching have learned a lot. And I'm just going to mention to everyone, please, Carlin put our survey in the chat. And so be sure to complete the survey, because that allows us to not only, what you're interested in, but it also helps us keep funding so we can continue to do this. We can encourage individuals to donate to SCM Link, and we can encourage corporations and foundations to continue to give us money to offer this programming to you. And then it lets us know feedback of your experience. And so before we wrap it up, I am going to have Carlin talk about the next things that are coming up this month for our programs.
Yes. So if you really enjoyed this cafe, then you will be thrilled to know that we are offering a 2 for this month. meaning that in our next cafe, that will also be on DNA. And that will be in 2 weeks, October 27th, same time, 6:30 to 7:30 p.m. Eastern. And our other program for October is our STEM in the City. And in that workshop, you will hear about how scientists in earth and environmental science use data visualization to study our Earth. And the speaker for that cafe will be sharing a really cool tool that will essentially help us to travel those different parts of the world that you can't get to by car or plane. So if you're interested to learn more, to also be a part of those cafes, then the link is in the comments in the chat.
Thank you so much, Carly. And if you have missed any of our previous cafes, be sure to check out our YouTube channel. We have lots of different STEM professionals that have shared their journey, and I'm certain that you may find one that piques your interest, even if it's not the same exact thing that you may want to pursue, but it may be close enough to help you do some research to guide. So thank you again, Dr. Phillips, for teaching us about your journey and about zebrafish as a animal model for disease research, but also letting us know, exactly when I heard you were a neuroscientist, I assumed the brain, right? And learning, that neuroscience is a very diverse field, right? And you can find something that you're interested in. So thank you everyone for joining us, and we will see you at our next event. And until then, just follow us on social media to stay abreast and find out what's going on with our programming. Have a great rest of your day.